globular proteins that work as a catalyst by speeding up chemical reactions without their structure being altered themselves. And within enzymes, substrates are the substances that enzymes convert into products. Enzyme substrate specificity involves substrates binding to a special region on the enzyme surface called the active site. The polar regions of the amino acids attract the substrate to the active site, and once the substrate collides with the active site in a correct orientation, it ends up staying there and catalyzes the reaction. The shape and chemical properties of the active site are specific to the substrate, so substrates will only bind to their specific enzymes. And then once the substrate is in the active site, the substrates are converted into products while they're bound there. And then the products are then released and the enzyme is used again by another substrate. Enzymes only work when the substrate collides with the active site at the right orientation. The movement of substrates and enzymes when dissolved in water or other fluids is completely random. So with that movement, there are collisions happening between the enzymes and the substrates, and the likelihood of the two colliding at the exact orientation in order for the enzymes to bind with the substrate and catalyze the reaction is actually very minimal. But the enzyme activity can be increased. So first, by temperature. So temperature affects enzyme activity in two ways. The first way is when a liquid is heated, its kinetic energy also increases, which leads to an increase in enzyme activity because the substrates and the enzymes are moving faster in random motion at a higher temperature, which leads to the increased likelihood of more successful collisions with the active site, which increases enzyme activity. However, the second way that temperature interacts with enzyme activity is when the temperature is too high, more bonds in the enzymes are broken because enzymes are proteins, and it causes the denaturation of the enzymes, which decreases enzyme activity. And there's an optimal pH for enzyme activity, and after that temperature has been reached, enzyme activity quickly decreases once that temperature exceeds the optimum temperature because the denatured enzymes cannot catalyze substrates anymore. pH also affects enzyme activity. The pH scale is used to measure acidity or alkalinity of a solution. So a lower pH on a scale of 1 to 14 is more acidic, compared to a higher pH is more basic. Enzymes have an optimum pH at which their activity is the highest, and if the pH is increased or decreased from the optimum pH, then enzyme activity decreases. This is because the hydrogen ion concentration that determines pH also has an effect on the enzymes, and if it is too high or too low, it affects the bonds of the enzymes and it denatures it. Finally, substrate concentration also has an effect on enzyme activity. Enzymes cannot catalyze reactions until the substrate binds to the active site. So if the substrate concentration is increased, then more collisions will occur and a higher number of successful collisions will occur. However, after the binding of a substrate to an active site, the enzyme is no longer available to other substrates until the reaction has been catalyzed. Therefore, enzyme activity remains relatively constant once substrate concentration has reached a certain point, and that's called the max rate of the reaction. It's like a situation where there's only a limited number of parking spots, let's say 20 parking spots. And originally you can add 3 cars and there's still a lot of available parking spots left. But if you add 20 more, now you have 23 parking spots. All the parking spots are taken up until one person leaves and the other cars can go in. So it's similar to that with enzymes. You can only reach a certain point, and then after that, the reaction rate just remains the same because there's no more enzymes to catalyze with the substrates. Enzymes can also be inhibited by other molecules, and there's 2 types of enzyme inhibition. There's competitive inhibition, which directly blocks the active site, so the substrate cannot enter the active site and be used by the enzyme to turn into products. But even with competitive inhibition, the same max rate of reaction will eventually be reached if more substrate is added because the enzyme numbers have not changed. Non-competitive inhibition, on the other hand, binds to another site, an allosteric site, on the enzyme, which alters the shape of the active site so substrates cannot enter and attach and catalyze with the enzyme. And non-competitive inhibition affects the max rate of reaction because it decreases because there are less enzymes available because the active site is temporarily changed so enzymes can't use it at all. Finally, immobilized enzymes are used in the industry because they are able to catalyze chemical reactions outside of living cells. The enzymes are attached to a material or aggregated so that their movement is restricted. The benefits of immobilized enzymes is that the enzyme can be easily separated from the products. Enzymes can be recycled and reused. Enzyme stability increases by immobilizing them, and faster reaction rates because substrates are exposed to a higher concentration of enzyme. And immobilized enzymes are used a lot in the biochemical industry, and examples of these in our daily lives includes in detergents, biofuels, medicine, paper, and even in the creation of lactose-free milk.